Now we are going to read chapter number 3rd of 11th NCERT Biology, that is Plant Kingdom. In the previous chapter, we look at the broad classification of living organisms under the system proposed by Whitaker, wherein he suggests the five kingdom classification with Monera, Protista, Fungi, Animalia, and Plantae. In this chapter, we will deal in detail with further classification within a kingdom Plantae, popularly known as Plant Kingdom. We must stress here that our understanding of plant kingdom has changed over time. Fungi and member of Monera and Protista have cell wall have now been excluded from plant through earlier classification placed them in the same kingdom. So the cyanobacteria that are also referred to as blue-green algae are not algae anymore. In this chapter, we will describe algae, bryophytes, pteridophytes, gymnosperm, and angiosperm under Plantae. Let us also look at classification within angiosperm to understand some of the concerns that influence the classification system. The earliest system of classification used only gross superficial morphological character such as habit, color, number, and shape of leaves, etc. They were based mainly on vegetative character or on androecium structure. Such systems were artificial. They separated the closely related species since they were based on few characteristics. Also, the artificial system have equal weightage to vegetative and sexual characteristics. This is not applicable since we know that the often vegetative character are more easily affected by environment. As against this, natural classification system developed, which were based on natural affinities among the organism and consider not only the external feature but also internal features like ultrastructure, anatomy, embryology, and cytochemistry. Such a classification of flowering plant was given by George Bentham and Joseph Dalton Hooker. At present, phylogenetic classification system based on evolutionary relationship between various organisms are acceptable. This assumes that organisms belong to same taxa have common ancestor. We know that us use information from many other sources to help. There is no support fossil evidence. Numerical taxonomy, which is now easily carried out using computer, is based on all observable characteristics. Number and code are assigned to all characteristics and the data are then proceed. In this way, each characteristic is given equal to important at the same time. Hundreds of characters can be considered. Cytotaxonomy, that is based on cytological information like chromosome number, structure, behavior, and cytotaxonomy that use the chemical constituent of the plant to resolve confusion, also used by taxonomists this day. 3.1 Algae. Algae are chlorophyll bearing simple thalloid autotrophic and largely aquatic, both marine and freshwater organism. They occur in various habitat, moist, stone, soil and wood. Some of them also occur in association with fungi, like in an animal, example in sloth and bear. The form and size of algae is highly variable, ranging from colonial form like volvax and filamentous form like Eurothrix and Spirogyra. A few of marine forms such as kelp form massive plant bodies. The algae reproduce by vegetative, asexual and sexual method. Vegetative reproduction is by fragmentation. Each fragment develops into thallus. Asexual reproduction is by production of different types of spore, the most common being the zoospore. They are flagellated spore and germination gives rise to new plant. Sexual reproduction takes place through fusion of two gametes. These gametes can be flagellated and similar in size as in Eulothrix or non-flagellated, non-motile but similar in size such as in Spirogyra. Such reproduction is called isogamous. Fusion of two gametes dissimilar in size as in species of Eurothrix is termed as anisogamous. Fusion between one large non-motile static female gamete and a smaller motile male gamete is termed as oogamous. Example: volvox and fucus. Algae are useful to man in varieties of ways. At least half of the total carbon dioxide fixation on the earth is carried out by algae through photosynthesis. Being photosynthetic, they increase the level of dissolved oxygen in their immediate environment. They are paramount importance as primary producer of very rich compound which form basis of all food cycle of all aquatic animal. Many species of Porphyra, laminaria, and Sargassum are among the 70 species of marine algae used as food. Certain marine brown and red algae produce large amount of hydrocolloids, example algin and carrageen red algae, which are used commercially. Agar, one of the commercial products obtained from Gelidium and Galaxcrian, are used to grow microbes and in preparation of ice cream and jellies. Chlorella, a unicellular algae rich in protein, is used as a food supplement even by space traveler. The algae are divided into three main classes: chlorophyce, pheophyce, and rhodophyce. 3.1.1 Chlorophyce. The member of chlorophyce is commonly called green algae. The plant body may be unicellular, colonial, or filamentous. They are usually grass green due to dominance of pigment chlorophyll A and B. The pigment are localized is defined chloroplast. The chloroplast may be discoid, plate-like, reticular, cup-shaped, spiral, or ribbon-shaped in different species. Most of the members have one or more storage bodies called pyrenoids located in chloroplast. Pyrenoids contain protein besides starch. Some algae may store food in the form of oil droplets. Green algae usually have rigid cell wall made up of an inner layer of cellulose and outer layer of pectose. Vegetative reproduction usually takes place by fragmentation or by formation of different type of spore. Asexual reproduction is by flagellated zoospore in Zoosporangia. The sexual reproduction shows considerable variation in type and formation of sex cell, and it may be isogamous, anisogamous, or oogamous. Some commonly found green algae are Chlamydomonas volvox, Eurothrix, Spirovara, and Chara. 3.1.2 The member of Pheophyce or brown algae are found primarily in marine habitat. They show great variation in size and form. They range from simple branch filamentous form to profusely branched form as represented by kelp, which may reach a height of 100 meter. They possess chlorophyll A, C, carotenoid, and xanthophyll. They vary in color from olive green to various shade of brown depends upon amount of xanthophyll pigment fucoxanthin present in them. Food is stored as complex carbohydrates, which may be in form of laminarin or mannitol. The vegetative cell have cellulosic wall usually covered on outside by a gelatinous coating of algin. The protoplast contain, in addition to plastid, a certainly located vacuole and nucleus. The plant body is usually attached to substratum by holdfast and has stalk. The stipe and leaf-like photogenetic organ, the fold. The vegetative reproduction take place by fragmentation. Asexual reproduction in most brown algae is biflagellated zoospore that are pear-shaped and have two unequal laterally attached flagella. Sexual reproduction may be isogamous, anisogamous, or oogamous. Union of gametes may take place in water or within oogonia. The gametes are pyriform and bear two lateral attached flagella. The common forms are Ectocarpus, Dictyota, Laminaria, Sargassum, and Fucus. 3.1.3 Rhodophyce. The member of Rhodophyce are commonly called as red algae because of predominance of red pigment are phycoceriothrine in their body. Majority of the red algae are marine, with generate concentrated found in the warmer areas. They occur in both well-lighted region close to surface of water and also great depth in ocean where relatively little light penetrate. The red thalli of most of the red algae are multicellular. Some of them have complex body organization. The food is stored as Floridian starch, which is very similar to amylopectin and glycogen in structure. The red algae usually reproduce vegetatively by fragmentation. They reproduce asexually by non-motile spore and sexually by non-motile gametes. Sexual reproduction is oogamous and accompanied by complete post-fertilization development. The common members are Phycosophinia, Porphyra, Glaciria, and Geludium. 3.2 Bryophytes. Bryophytes include the various moss and liverwort that are found commonly growing in moist shaded area in the hills. Bryophytes are also called amphibians of plant kingdom because this plant can live in soil but depend on water for sexual reproduction. They usually occur in damp, humid, and shaded localities. They play important role in plant succession on bare rocks and soil. The plant body of bryophytes is more differentiated than the algae. It is thallus-like, prostrate or erect, and attached to substratum by unicellular or multicellular rhizode. They lack true system or leaves. They may possess like root, like leaf-like structure, stem-like structure. The main plant body of bryophyte is haploid. It produces gametes, hence it is called gametophyte. The sex organ in bryophytes are multicellular. The male sex organ is called antheridium. They produce biflagellated anthrazoids. In female sex organ is called archegonium is flask shaped and produce single egg. Anthrazoids are released into water where they come in contact with archegonium and anthrazoid fuses with egg to produce zygote. Zygote do not undergo reductional division immediately. They produce a multicellular body called sporophyte. The sporophyte is not free living but attached to the photosynthetic gametophyte and derive nourishment from it. Some cells of sporophyte undergo reductional division meiosis to produce haploid spore. These spores germinate to produce gametophyte. Bryophytes in general are little economical importance, but some mosses provide food for herbaceous mammals, birds, and other animals. Species of sphagnum and moss provide peat that have been long used as fuel and packing material for transshipment of living material because of their capacity to hold water. Moss along with lichen are first organism to colonize rock, hence are the great ecological importance. They decompose rock making the substrate, the suitable for growth of higher plant. Since moss form dense mat on the soil, they reduce the impact of falling and prevention of soil erosion. The bryophytes are divided into liverwort and mosses. 3.2.1 Liverwort. The liverwort grows usually in moist, shady habitats such as bank of stem, marshy ground, damp soil, bark of tree, and deep in the wood. The plant body of liverwort is thalloid. Example: Marchitania. The thallus is dorsiventral and closely appraised to substrate. The leafy member have tiny leaf-like appendages in two rows on the stem-like structure. Asexual reproduction in liverwort takes place by fragmentation of thalli or by formation of special structure called gamma. Gamma are green multicellular asexual bud which develop into small receptacle called gamma cups located on thalli. The gamma become detached from parent body and germinate into form of new individual. During sexual reproduction, male and female sex organ are produced either on same or different thalli. The sporophyte is differentiated into food seta capsule. After meiosis, spores are produced within the capsule. These spores germinate to form free-living gametophyte. 3.2.2 Mosses. The predominant stage of life cycle of mosses is gametophyte, which consists of two stages. The first stage is protonema stage, which develop directly from a spore. It is creeping, green, branch, and frequently filamentous stage. The second stage is leafy stage, which is developed from secondary protonema as a lateral bud. They consist of upright, slender axes bearing spirally arranged leaves. They are attached to soil through multicellular and branched rhizoid. This stage become the bears the sex organs. Vegetative reproduction in mosses is by fragmentation and budding in secondary protonema. In sexual reproduction, the sex organs antheridia and archegonia are produced at the apex of leafy shoots. After fertilization, the zygote develops into sporophytes consisted of a fruit, seta, and capsule. The sporophytes in mosses is more elaborated than in liverwort. The capsule contains spore. Spore are formed after meiosis. Meiosis have an elaborative mechanism of spore dispersal. Common example of mosses are Funaria, and Polytrichum, and Sphagnum. 3.3 Pteridophytes. Pteridophytes include horsetail and fern. Pteridophytes are used for medical purpose as as soil blinder. They are also frequently grown as ornamental evolutionary. They are first terrestrial plant to possess vascular tissue, xylem and phloem. You shall study more about this tissue in chapter 6. The protopteridophytes are found in cool, damp, shady places, though some many flourish well in sandy soil condition. You may recall that in bryophyte, the dominant phase in life cycle is gametophyte plant body. However, in pteridophyte, the main plant body is sporophytic, which is differ into true root system and leaves. These organs possess well differentiated vascular system. The leaves in the pteridophytes are small as in Selagella and large as in fern. The sporophytes bear sporangia and are covered by leaf-like appendages called sporophyles. In some cases, sporophyll may form distinct compact structure called strobili or cones. The sporangia produce spore by meiosis in spore mother cell. The spore gametes to give rise to inconspicuous but small multicellular free-living, mostly photosynthetic thalloid gametophyte called prothallus. These gametophytes require cool, damp, shady places to grow. Because of this specific restricted requirement and need for waste water for fertilization, the spread of living pteridophyte is limited and restricted to narrow geographical region. The gametophyte bear male and female sex organ called antheridia and archegonia, respectively. Water is required for transfer of arthrozoites. The male gamete relays from antheridia to the mouth of archegonia. Fusion of male gamete with the egg present in archegonium result in the formation of zygote. Zygote thereafter produce multicellular well-differentiated sporophyte, which is dominant phase of pteridophyte. In majority of the pteridophytes, all spore are similar kinds. Such plant are called homosporous. Genera like Silaginella and Salvania, which produce two kinds of spore, macro and micro spore, are known as heterosporous. The megaspore and microspore germinate and give rise to female and male gametophyte respectively. The female gametophyte in this plant are written on parent sporophyte. For various period, the development of zygote into young embryos take place within the female gametophytes. This event is precursor to the seed habit. Considered an important step in evolution, the pteridophytes are further classified into four classes: Philospida, Lycospida, Spinatspida, and Pteridospida. 3.4 Gymnosperms. The gymnosperms are plants in which ovules are not enclosed by any ovary wall and remain exposed both before and after fertilization. The seeds that develop post fertilization are not covered, that is, are naked. Gymnosperm include medium-sized trees or tall plant and shrubs. One of the gymnosperm tree, the giant redwood tree sequoia, is one of the tallest tree species. The roots are generally taproot. Roots in some genera have fungal association in the form of mycorrhiza, while in some other small specific roots called caroid root are association with nitrogen-fixing cyanobacteria. The stem are unbranched cycles or branch, pinus or caterus. The leaves may be simple or compound. In cycles, the pinnate leaf persists a few years. The leaves in gymnosperm are well adapted into withstand extreme of temperature, humidity, and wind. In conifer, the needle-like leaves reduce the surface area. Their thick cuticle and sunken stomata also help to reduce water loss. The gymnosperm are heterosporous. They produce haploid microspore and megaspore. The two kinds of spore are produced within a sporangia that are borne on sporophyll, which are arranged spirally along an axis to form lax or compact strobili or cones. The strobili bearing microsporophyll and microsporangia are called microsporangiate or male strobili. The microspore develop into a male gametophytic generation which is highly reduced and is confined to only limited number of cells. This reduced gametophyte is called pollen grain. The development of pollen grain takes place within the microsporangia. The cone-bearing megasporophyll with ovule or megasporangia are called microsporangiate or female strobili. The male and female cones or strobili may be borne on same tree, pinus. However, in cycles, male cone and megasporophyll are born on different tree. The megaspore mother cell is differentiated from one of the cell of the nucellus. The nucellus is protected by envelope and composed structure is called an ovule. The ovule are born on megasporophyll which may be cultured to form female cone. The megaspore mother cell divided meiotically to form four megaspore. One of megaspore enclosed within the megasporangium develop into multicellular female gametophyte that bear two or more archegonia or female sex organ. The multicellular female gametophyte is also retained within megasporangium. Unlike bryophyte and pteridophyte in gymnosperm, male and female gametophyte do not have independent free-living existence. They remain within the sporangia written on sporophytes. The pollen grain is released from the microsporangium. They are carried in air current and come in contact with opening of ovule, born and megasporophyll. The pollen tube carrying the male gamete grows toward archegonia in ovule and discharges their content near the mouth of archegonia. Following fertilization, zygote develop into embryo and ovule into seed. These seeds are not covered. 3.5 Angiosperm. Unlike gymnosperm, where the ovule are naked, in the angiosperm, the flowering plant, the pollen grain and ovule are developed in specialized structure called flower. In angiosperm, seeds are enclosed in fruit. The angiosperm are exceptionally large group of plant occurring in wide range of habitat. The range size from smallest wolfia to tallest equinocyphytus. 100 meter. They provided us fodder, fuel, medicine, and several other commercially important products. They are divided into two classes: the Dictya, dicotyledon, and monocotyledon. The dicotyledon are the characterized by seed having two cotyledon, reticulate venation, leaves, and tetramerous and pentamerous flower. That is having four or five member in each floral whorl. The monocotyledon, in other hand, are characterized by single cotyledon seed, parallel venation in the leaves, and trimerous flower having three members in each floral whorls. The male sex organ in flower is the stamen. Each stamen consists of a slender filament with an anther at the tip. Within the anther, the pollen mother cell divided by meiosis to produce microspore, which matures into pollen grain. The female sex organ in flower is pistil. Pistil consists of ovary at its base, a long slender style and stigma. Inside the ovary, the ovules are present. Generally, each ovule has a megaspore mother cell called undergoes meiosis to form four haploid megaspores. There three of them degenerate and one divided from embryo sac. Each embryo sac has three cells called agapparatus, one egg cell and two synergids, three antipodal cell, two polar nuclei. The polar nuclei eventually fuse to produce diploid secondary nucleus. Pollen grain, after dispersal from the anthers, are characterized by wind and various other agencies to the stigma of pistil. This is termed as pollination. In pollen grain germinate on the stigma and result pollen tube grow though through the tissue of stigma and style reach the ovule. Pollen tube enter the embryo sac where two male gametes are discharged. One of male gamete fuses with egg cell to form zygote. The other male gamete fuses with diploid secondary nucleus to produce the triploid primary endosperm (PEN). Because of occurrence of two fusion, that is syngamy and triple fusion, this event is termed as double fertilization, an event unique to angiosperm. The zygote develop into an embryo and the PEN develop into the endosperm, which provides nourishment to developing embryo. The synergids and antipodals degenerate after fertilization. During this event, the ovule develop into seed and ovaries develop into fruit. The life cycle of an angiosperm is shown in figure 3.6. 3.6 Plant life cycle and alternation of generation. In plant, both haploid and diploid cell can be divided by mitosis. This ability leads to conformation of different plant bodies, haploid and diploid. The haploid plant body produce gamete by mitosis. This plant body represent gametophytes. Following fertilization, zygotes are also divided by mitosis to produce a diploid sporophytic plant body. Haploid spore are produced by the plant body by meiosis. This in turn divided by mitosis to form a haploid plant body once again. Thus, during the life cycle of any sexual reproducing plant, there are an alternation of generation between gamete producing haploid gametophyte and spore producing diploid sporophyte. However, different plant group as well as an individual represent them different in the following pattern. First, sporophytic generation is represented by only the one-celled zygote. There is no free-living sporophyte. Meiosis in the zygote result in the formation of haploid spore. The haploid spore divided meiotically and form gametophyte. The dominant photosynthetic phase in such plant is free-living gametophyte. This kind of life cycle is termed as haploentic. Many algae such as volvox, Spirogyra, and some species of Chlamydomonas represented this pattern. Second, on the other extreme is the type where the diploid sporophyte is dominant, photosynthetic independent phase of plant. The gametophyte phase is represented by single to few cell haploid gametophyte. This kind of life cycle is termed as diplontic. An algae fucus Spirogyra represent this pattern. In addition, all seed-bearing plant, that is gymnosperm and angiosperm, follow this pattern with some variation, wherein the gametophyte phase is few multicelled. Bryophyte and pteridophyte interestingly exhibit an intermediate condition, haplodiploidy. Both phases are multicellular, however, they differ in their dominant phase. Dominant independent photosynthetic thalloid R or erect phase is represented by haploid gametophyte and its alternate with the short lived multicellular sporophyte totally or partially depend on the gametophyte for its arch anchorage and nutrition. All bryophytes represent this pattern. The diploid sporophyte is represented by a dominant independent photosynthetic vascular plant body. It's alternate with multicellular saprophytic autotrophic independent but short-lived haploid gametophyte. Such a pattern is known as haplodiplontic life cycle. All pteridophytes exhibit this pattern. Interestingly, while most algae genera are haploidic, some of them such as Ectocarpus, Polycympia kelp are haplodiplontic. Fucus and algae is diplontic. Thank you.